Hello everyone, welcome to the Wise Tutor YouTube channel. Today's video is video number 6 in the AP Biology sequence. We've got the fourth macromolecule, which is nucleic acid, and this is our biochemistry unit. So, for nucleic acids are responsible for the storage and expression of genetic information or material. And there's definitely a whole unit of biology that we're going to get into in the future that is dedicated to nucleic acids and DNA and RNA. But basically, just know that this is just maybe sneak peeks into the future unit. The monomer for nucleic acids is the nucleotide, and we'll see the structure of a nucleotide. Just like the amino acid of a protein has a specific structure, the nucleotide also has a specific structure with different components. And we'll see that on the next slide. The polymer is nucleic acids, and the bond is called the phosphodiester linkage. And so you see a picture of that here. You got one nucleotide right here, and it is connected by a phosphodiester bond here, and here's the bottom one is also a nucleotide. And one of our future videos will be addressing these two processes that allow us to build up multiple multi-monomer structures, so polymer structures like this from their monomers, their individual monomers. You'll see that in a very future and very upcoming video. Nucleotide structure. So the nucleotide contains a phosphate group. Here is the phosphate group. It's the center of it is a phosphate. A pentose sugar, which is shown here. It's basically a carbon ring like we discussed for carbohydrates. And there are two variations. There's a deoxyribose variation in DNA and a ribose variation in RNA. As for the third component, this one right here, this is what we call a base pair or a nitrogenous pair, base. And we have two kind of classes of these base pairs. We've got pyrimidines, which have a structure of single rings, and we've got purines, which have a structure of double rings. All right. Cytosine and thymine are the two base pairs in DNA. However, in RNA, you've got cytosine, but instead of thymine, you replace it by uracil. Right, and we'll get to that obviously in the future unit. But this is also important to know here. Purines, the double ring compounds, we've got adenine and guanine, and those stayed exactly the same. Now, another important term to know is nucleoside. A nucleoside is basically a nucleotide, so it consists of these, without the phosphate group. So if you subtract the phosphate group, you have a pentose sugar and a base pair. And that's exactly what a nucleoside is. You see it, it's almost as if this part didn't exist in the diagram at all. We could cross that out, and you've got this structure right here being a nucleoside. And so I just wanted to show a bit more pictures of what is really part of the structure of nucleotides and how they play a role in DNA and RNA structure. And so you've got cytosine and guanine that are attracted to each other. Cytosine and guanine, these. So usually, and not usually, always, one purine is attracted to one pyrimidine, and it's always the same one. So cytosine is always attracted to guanine. You'll always see those being paired up together. Adenine is always attracted to thymine. You'll always see those paired together in a DNA strand or a double strand. But in an RNA strand, you'll see the adenine being attracted to not thymine, but uracil instead. We don't have a picture of uracil here, but it looks very similar to thymine as well. Now, in terms of actually seeing it, in format of DNA strands, you could pretend that this goes down forever, this structure goes down forever, and it creates a strand of DNA. And the same thing could happen for this side as well. You can see a full strand of DNA on one side and then the other side. Guanine, as I said, is always going to be attracted to cytosine. So here's the single ring that we were talking about that characterizes pyrimidine. And you've got guanine, which is a purine with two rings, all right, a double ring. This, and what actually attracts them, and this is really important to know, I should have labeled this in the slideshow. What attracts the cytosine and guanine, or kind of, pairs them up together, are hydrogen bonds. All right, so these dashed lines here are hydrogen bonds. So already from the first couple, lectures in the biochem unit, you could see how important hydrogen bonds are for biological processes. They hold base pairs together when you're making DNA, and that's really important. And you'll see that in also the future unit. The same thing happens with thymine and adenine. And their attraction. They are attracted by hydrogen bonds here as well. And so you've got the double ring of adenine, which again is a purine, and you've got a pyrimidine, thymine, that is a single ring, and they're attracted via hydrogen bonds. Now, one more thing that could help you remember or distinguish purines and pyrimidines is the Y in pyrimidines matches with the Y in cytosine and the Y in thymine. And that's a really good kind of like way to memorize what the pyrimidines are. Now all you need to do is associate that pyrimidines have a single ring structure, and they C's and T's. Purines have a double ring structure, and they are adenine and guanine. And always remember, you'll never see an adenine and guanine being attracted to each other. You'll never see a cytosine and thymine being attracted to each other. It must be a purine being attracted to a pyrimidine. All right, it cannot be two of the same are attracted. It has to be opposites attract, almost kind of like a charge. And so I hope you enjoyed this video about the nucleic acids. It is our fourth macromolecule. We are getting super close to finishing the biochem unit, and I'll see you guys in my next video.